as an organizer, and I'm really very happy to be here. I mean, it's a real pleasure to be with you. I hope I can offer, let's say, at least a flavor about the molecular part, I mean, the molecular simulation part, and try to integrate it with the other pieces that you have been seeing during the last days. So the first thing that I would like to do is, come on, now we have AlphaFold. You heard about AlphaFold during the last 4 or 5 days, at least here. Some people already have been even working with it before. And what about simulations? I mean, do they still have a place now? And in particular, I mean, in my area, like since we develop and apply constant pH simulation methods, do you still need them? Well, of course, I mean, if you look to the literature, you can see that the protein revolution has already, I mean, we are in the middle of this process, I should say, because it has started at least 5 years ago. AlphaFold has completely changed the field. It has been frustration for some people. It has been maybe the wrong idea in my eyes that we don't need more experimental data. But for sure, we have to assume that it has been really a big step forward. I mean, we are in another level. You can see this, that the old problem that people were trying to solve during many, many years. I mean, let me see if I can find it. So giving a structure, or sorry, a sequence, now we can predict the 3D. Oops, I'm getting troubles with this guy. It's not working. Yeah, now it is working. Okay. So now you can see that we found a way. So the basic idea here is, for most of the biological people, what would be important is just to get or to predict the 3D structure. It doesn't matter how you do it. I mean, you could find someone outside that I say, hey, I have this sequence. Can you give me the coordinates of my protein in the 3D space? And that would be the problem. So you can see that even a crystal ball could make it. But they found that AlphaFold can be quicker and efficient. So this is one of the goals. We can go on, I mean, with this revolution. I mean, we can say, I mean, there are just different papers that were published and discussed this in the literature during the last at least 5 years, as I said. But we can go further, and it's not just the revolution in terms of biology, but also you can see that's going to be affecting neighbor business, like medicine. And if we go even further, we can see that it's also affecting chemistry, the pharmaceutical science, and so on. So this is clear. Probably as a good example, the Nobel Prize that you already saw in many slides during the last days. Just remind you, we are in a physics institute. So it's good to remember that it was thanks to physics that all this revolution has appeared. So I just try to remind you, and then I make a connection with what Rolf offered you during the last days. So you had like the basic understanding that's really important to know what you are doing. Otherwise, all these tools, they just happen to be a black box that you try to operate. So we have to be careful to remember that there are, let's say, fundamental things. And one of the things that I'll try to do to you also today is to try to go back to terms or quantities that were presented here in the school and remind you from the basic science what do they mean as well. So this is, let's say, the overview. And at this point, you can start asking yourselves, how good is it? I mean, is it really like it has solved all the problems? So now we have to try to find other jobs. I mean, we change our business because it's over the problem. And in fact, I mean, the physics is still remaining a big challenge in these things. And I will try to show you a couple of examples for you like that. Of course, you have database. I mean, you can get structures for a huge number. You probably remember when Chuck was here that he was describing to you the number of available structures on the PDB that are coming from computational studies. If I'm not wrong, he gave a number about 1 million, right? Do you remember it? It was on the beginning of the week. So we have really a nice database. But first of all, I would like to make a very clear difference between two different problems. So one is simply to predict the 3D structure. So giving a sequence, you get the 3D structure. A very similar problem, but completely different by one other side, is a protein folding. So AlphaFold has not solved the protein folding problem. I mean, if would you like to understand the mechanisms, how a protein is going to go to this place and understand what is happening between, you don't have this answer from AlphaFold. And one reason for it is because we are still having some troubles with the force fields and how we solved our modulus. So if you're coming from physics, maybe I can tell you that your Hamiltonian, you were joking yesterday, or your model, I mean, your force field is essentially your Hamiltonian in physics. And in this case, this is still an open problem. But it's not all. Let me go further. So instead of looking to proteins, let's look to something much simpler. Let's look to peptides. So in this case, you can see that we have a small peptide here, and we try to benchmark pKa's. Soon, in a couple of slides, I'm going to go back and explain what does a pKa really means. But in this case, we have done a couple of different things. I mean, this is a way to assess if the quality of your electrostatic predictions are okay or not. Don't worry, I'm come back to all the details in a couple of minutes. This is like just to show you where we are and to try to motivate why I needed to go later, to continue with the physical part. So the pKa's are used to benchmark to assess if the electrostatic properties of your model are correct. So if you're describing nature as it should be. And you can see that I have data for a couple of molecules here, a couple of peptides. So here are the amino acids that can titrate, so they can change their protonation state. We have the experimental data from NMR. You probably remember a bit about it from Monday. We have what we call the null model. The null model is if you assume an ideal behavior. So you don't have interactions between the neighbors. So you just look to an isolated amino acid, which is not true, and I'm going to show you also this part when they are in a 3D structure, because you have neighbors that can also have charges. So they can also affect each other, right? And here we have results from our Altamus simulations, coarse-grained models that are flexible, and you have results using one of the titration models that we call fast proton titration scheme. I'm going to detail this to you in the next class. And in this model, it's a rigid model, so we needed to use, to provide the model, the 3D structures of the peptide. And we have a different source. I can just create it by random, by scratch, I can create a peptide structure. This is essentially what you are doing if you're using PyMol. You can use AlphaFold to predict it as well. Just to give it a sequence, it's a small sequence, it's going to be even quicker. You get an answer, you get an ensemble of confirmations if you'd like. You can also use another tool that was specifically designed for peptides. So it's called PepFold. There are 4 versions of it. The last one can even incorporate pH effects. So it's nice. And then, of course, you can compare the other techniques. I'm not going to all the details, but just to show you the quality of the results. Any model that you want should be better than the new model. The new model is more or less, you don't even need to do a math. If you know the ideal behavior, you assume an ideal behavior, and you are done. You can calculate this by hand. So if the model is not better than the new model, which means that the model is not, what is the point to run a simulation if you can take a paper and get a good result, or even better than this one? So we do like to be better, and you can see the numbers here. There are descriptors like you calculate the maximum average deviation, you calculate the RMS, the root square mean deviations, you calculate the maximum number, the average. You can do different metrics for it. So you can get, in some way, the quality of your prediction. And if you look at the new model, I have here 0.24 for the RMSE. So the root mean square error estimated for this guy is 0.24. When you look to the results that you get from AlphaFold, you can see that is 0.29. So the structure that AlphaFold provides you for a peptide is worse than what you're going to get if you just do something random. You can see that the number is essentially what you get from PyMol. So if you, by scratch, you make the confirmation of your peptides, you're not going to get something better than the new model. And the AlphaFold is going to behave exactly in the same way. So this is something that starts to worry you. Come on. Depending on what molecule you're using, how far we can go now with my peptide, increasing the number of monomers, so I start to go back to a good prediction. So this is just one example for you to keep in mind about these things. And actually, the best result, as you can see here, is where you can get either using our Altamus simulation or you use AlphaFold. 0.21, now you're doing better than the new model. Okay, this is not all. If you work like me with antigen-antibody interactions and you try to use AlphaFold for this specific class of molecules, you can see some troubles. So now we're talking about AlphaFold 3, and in this case, you can see that the interface has the higher trouble. I mean, it's where you have large deviations. So it's hard to get a good prediction of the interface. That, by the way, it's what you were after, right? Because it's typically what matters more for most of the people that are studying these things. They would like to well characterize the interface. So AlphaFold can give you some troubles to predict well. There are other tools, I mean, similar tools as AlphaFold, specifically designed for antibodies. IgFold is one example. There is ABB Builder. There are different tools that can be a little bit better for antibodies. In fact, if you remember what Elder said yesterday, the pharmaceutical companies are already using their own data banks to create their own AlphaFold versions and probably incorporate better and they use a better train so the results can be better. But, I mean, the original version of AlphaFold can provide you this kind of data. Even so, if you use such things, you can see trouble also in the position of the interface. I mean, here you can see how it looks like the receptor binding domain from SARS-CoV-2 and how you're going to predict its position using AlphaFold. And again, the interface can be misplaced if you compare with the original antibody that's available. So you can see that the orientation is also a little bit out. There are more critical examples. This is one of my favorite ones. Some proteins, they have been crystallized under different conditions. The dengue envelope protein, the E protein, is a good example. If you go to PDB, you have this PDB ID here that tells you how is the structure in the pre-fusion. You also have the post-fusion that was crystallized in pH 7. The release data is slightly different, so it could help you in some way when you are using AlphaFold. In particular, you can put a criterion that you just use the database up to 1 point, so eventually you could play with it. But in fact, when you're running AlphaFold now, if you give it a sequence and you run AlphaFold, what are you going to get? Well, it turns out that you only get one of them. You don't get the other. So if I run AlphaFold for the same sequence, I mean, of course, they have the same sequence, and then if you make a comparison, you can see that 100% of your time, you are getting just the one that was from pH 80.5. So if your application, when you're running your calculation, you were trying to target or to reproduce experimental data that was done at pH 7, you are not using the right confirmation. This is a real problem. So these things are telling you that, well, we still need simulation. And in particular, I mean, if you'd like to address pH effects, you definitely need constant pH simulation. So I hope with these examples, I could show you a little bit, I mean, why we still have the need for pH effects. Just imagine, I mean, one example, if you would like to go for a more complex protein, like insulin, you have crystal data from pH 7 to 11. So there are a couple of structures on PDB with all, I mean, that were crystallized under all these different conditions. And again, when you get a result, an ensemble result from AlphaFold, how do you know which one corresponds to what pH condition do you want? You are simply going to pick up one. They can be quite different. I mean, I'm trying to show here separation distance between different groups of the proteins. And you can see that in different pH, their separation distance is quite large. So the gate can be open or the gate can be closed, depend what structure you are using. And how do you know, before doing anything, which one is the appropriate one for you? So these are points that I'd like you to keep in mind. I mean, and this is actually the motivation why I'm going now to the simulation part. But they are really important. I mean, if you'd like to address problems like if you're working, for instance, with cancer, well, one thing is that you have to be worried in cancer is to avoid that these two proteins here, they interact with the DNA. And this is a problem that's controlled by pH. So in this case, if you use AlphaFold, you're never going to pick up this phenomena. Is that clear? Am I too fast? Can you follow me? Are we with me? Is everybody, I mean, did you have a very good breakfast? Yes, please, Pedro. Not really.
Why is that? Because, well, normally you get similar, you are more interested in physiological pH. So,
Ah, you mean pH 7? I'm not sure. Depends how was the bias. I mean, that's actually something that we can ask Elias because he has been looking to the pH distributions. And I think pH 7 is the one that appears often, but you can double check with him. Because this is an analysis that he's doing. Yeah, I mean, he's looking to data banks and trying to see for the crystallization, I mean, the structures that are available, how is the distribution of the pH. But I'm not sure, I mean, how this information has really been impact the final results. Like in the case that we just saw for dengue enveloped protein, it was not true. If you remember what I showed, I mean, the structure that appears more often in the example provided by not all often, I mean, it was the only one that appeared on AlphaFold was the pH 80.5. So for some structures, somehow AlphaFold gives you the structure that is not even in the prop. So I can take what you asked and make another statement. This can be even more dangerous than what I'm saying. Because you see, the common way of us to think is, okay, it's going to be pH 7. But in fact, I got the one that was more, I mean, the one that was from pH 80.5. Yes, please.
do you think that the information about which case can be implicit in the model, but maybe by, for instance, running the full approach, maybe you cannot see this, and when, because people, I saw some people working on the MSA, different MSA strategies, different sampling strategies to try to find other formations of those proteins. Do you think that the information is there, but you need to do some exploration?
I think it's missing information. That's why my honest opinion is that we need to combine the physical models with machine learning. I mean, it's only this combination that probably can compensate the lack or the bias that's there. For instance, most of the data is crystallograph data. And to crystallize a protein, you need to do whatever you need to make sure that the two proteins are coming together. So often the conditions that they are running the crystallization experiments, they are not the ones that you have in physiological conditions. So this can be one problem. And this is going to be biased in your prediction. So one way is probably that we find another source of data that can be more reliable or in some way can compensate what's missing there and try to incorporate. But, I mean, it's still an open problem. I mean, there are, let's say, strategies, for instance, that you can improve the results for fold for sure. But I don't think just the machine learning part can make the trick. I don't think we have enough data. And there's also a risk now that less people were going to work with experimental to get more data. It's because people are going, come on, why am I going to crystallize a protein if I can just go to my laptop, go to the homepage of AlphaFold, and get a couple of examples of my structure. So depending on what you are after, this could be enough. And on the older days, it was needed, I mean, even for simple things, I mean, to do the crystallization and all the process to get the 3D structures. So for me, I mean, it is still the need of more experimental data, more physical-based simulation, and the combination of these things with the machine learning that can probably give us the next level of information. Anybody, I mean, would like to make another question? I mean, feel free to raise your hands. I mean, I'll try to be more relaxed, and even the time, I mean, if I don't have time to finish everything that I had in plan here, we can continue later anyway. If not, let me go. So let me start. I mean, I was talking about the cancer just as an example, but now let's see. I mean, how we start to build up. I mean, or to collect all these pieces. So biomolecular interactions. When you think about biomolecular interactions, we would like to do a couple of things. I mean, giving a protein and giving a polyelectrolyte. There are a couple of things I would like to know. First, if they are going to form a complex. If they do, in what conditions? And here it comes also the pH in the game. And we also like to answer the question if the complex is a stable one. We'd like to know what are the critical amino acids for this process to happen, or what kind of replacement, what kind of mutations we can make to improve the binding affinity, for instance, examples that you saw yesterday. So how we can engineer better molecules if we make a replacement of amino acids. That's what comes here. And coming from physics, there's another thing I'd like to ask yourself. Come on, what is the driving force? So it's not just to predict what is the best molecule, but why and what is happening in terms of physics, that one is the best molecule. Because back to what I just replied, if you know these answers, you can think about better models. You can also think about how you can use more of this information. So your physics intuition is quite important here. And just to give an example, I all the time tell people that proteins are like humans. They have a personality, and everyone is different. So you can see in nature that some proteins, they like to interact using charge-charge interaction. They like Coulomb interactions. Others, they like to be like ion-dipole interactions. And this is typically what we study in most of the courses. But we also have what we call mesoscopic forces. And it's here that we can see very, very nice things. For instance, you can see the complexation of a negative polymer with a negative protein, or a negative protein with a negative protein. You can take RNA and a protein, and under conditions that they have the same net charge, they attract each other. And come on, you are going to say now, come on, when I was in basic class, I heard about this Coulomb law. And according to the Coulomb law, they should repel each other. But how on earth they attract each other? I'm going to give you these examples tomorrow. But let's start from the beginning as an example. The Coulomb interaction. One nice example of the Coulomb interactions is the SARS-CoV-2. So SARS-CoV-2 is very nice to study physics, by the way. And it happens to be our last school here in 2020 that we have been presenting data at the very early days of the pandemic about the systems without experimental data. I'm also going to show you tomorrow these things, so wait and see. But I can anticipate a couple of things. I mean, the interaction between the SARS-CoV-2 RBD, the receptor binding domain, and the cell receptor AC2 is a good example of charge-charge interaction. So if you look to this guy here, here you have the receptor binding domain, you have the AC2 that we have in our cells, and that's the entering door of the virus. If you're not familiar with it, just in case, I have here the spike protein, I have the receptor binding domain on the top of that. This is probably the picture that has appeared over the years. Probably is one of the most popular pictures in the news. And in this guy, we can see that the binding affinity is a function of charge that also happens to be a function of time. This is nice. You can see that the virus is able to learn physics. So as the time is passing, we are having more and more charges on the molecule. And it starts to be more and more attractive. By the way, if you take 5 points in this plot, doesn't matter which variant you pick up, with 5 points, you can get a linear regression, a very basic machine learning idea, I would say the basic one, and doing so, you can predict what is coming next. So just to give an example to you, when we did this work in 2022, this variant here, XBB1.5, was not available. You didn't know about it. So 1 year later, more or less, when it appeared, we test the model, and we can see that it's following the same rule. So it's possible to predict how the virus is evolving looking to electrostatics. I'm going to show you more examples tomorrow. So this is just now to illustrate that charge-charge interactions can be important. And the RBD is one example of the molecules that behave with this property. So there are happy people, unhappy people, there are different personalities. This is one example of one personality of the proteins that they like to interact with others using charge interactions. This is one example. Just one thing that is important here, as I can see, and I should forget, cannot forget to tell you, is this dependence. I talked about charge, but you have to remember that charge in a protein is a function of a pH. Come back to this point a couple of times today, but keep this in mind. Another example of ion-dipole interactions you can find in biomaterials. This used to be my favorite example, so if you go back to the YouTube link of the school in 2020, I probably gave lots of examples about this molecule because it was one of my favorites 5 years ago. And in this case, we have a very strong material, the spider silk, and here if you look at this protein here from that makes the spider silk, where you have this red circle here, is where you have the interactions of the N-terminal domain of this spider protein, and they form this very strong material that happens to be controlled by pH. So under pH around 7 and 8, you have the monomers. When you start to make more acid, they start to aggregate and they form this strong material. So if you see a spider somewhere, drop some acid material, and because if it's go below 3, you have something that they're going to make them apart. So you can destroy the spider silk using either something that's very acid or very basic. There are products about this in the market. I mean, I'm not going to go to all details, but just let you know, I mean, this is the example that I was saying. I mean, you have here pH 7, they are monomers. Then if you go to pH 6, they form a complex. So it's controlled by pH. You can imagine lots of medical applications. This is why we already have this BioSTU. And one point of this, of course, was to understand the protein, what was the driving force, and also to try to predict how you can shift the pH conditions using mutations where it can form a better, or you have a stronger binding affinity. But just to go back to the point, I mean, when you try to understand why these proteins, they bind to each other, you can see that the driving force is here. Oops, sorry. I'm getting troubles with this pointer here. Anyway, it's here. And the reason why they form a complex is thanks to the dipole interactions. So this is another example. I mean, I showed you the RBD as charge-charge interaction. Now I'm showing you another example that happens to be ion-dipole interactions. Okay? And again, the dipole is also a function of pH. So all the quantities that we often have to explain the interaction of the proteins, they happen to be dependence of pH, something that's absent in AlphaFold, at least so far. So now that I probably have convinced you that's interested to know a little bit more about constant pH methods, I mean, I talk about pH all the time. So let's go further and try to see how I'm going to make the structure of this set of 3 lectures. So I'm going to walk you using people that are probably familiar from you, I mean, from Boltzmann to Coulomb. And the general context, I mean, is what I just have done. The next step is now I'm going to introduce what exactly we do in our lab. And I'm doing so, I'm also going to give you one of the possible strategies to work in this field. So this is also part of, let's say, of the class. I'm going to use my own example as a way to show you a couple of things. Next, we go very basic. I mean, I know that there are people coming from biological science, there are people coming from computational science in different fields. So I'm going to try to pinpoint a couple of, let's say, key concepts in physical chemistry. For instance, I'm going to introduce to you what is a pKa. I mean, we all talk about pKa, and there are people that often, I mean, we meet during a coffee, what was a pKa? So what is this? What is pKa? So I'm going to try to brief this. I hope you don't get too much bored. I mean, if you are already in the field, maybe there is something new. But I also need this part to introduce you and to explain how we have developed FFPS. So to come to FFPS, I need to go back to this basic physical chemistry. And also to show you a couple of other examples there. Next, I'm going to illustrate how normally we work with electrostatic interactions. Just citing something about the experimental data and also going to the simulation and from a historical point of view. That's something. And in terms of history, one important model was the Tanford-Kirchhoff model. This happens to be also the core of the FFPS. So most of the applications when we are dealing with electrostatic interactions, somehow we are using this historical model. So if you learn a bit of it, I mean, the main ideas, you are already prepared to work in the field. So that's one of the goals. More or less connected with it, I have to touch a bit about the Poisson-Boltzmann equation. And then, I mean, we leave this, let's say, basic part, I mean, the basic ideas, and we move to constant pH methods where I give you a flavor of what is available in the market. And for tomorrow, depending how much time we have or how much you are interested, we can go to more examples. So here is like, I'm going to describe this charge-charge interactions in the sense that one is negative, the other is negative, and they form a complex. What we call, I mean, it has been called in the literature, the complexation on the wrong side of the isoelectric point. So this is the plan, right? Well, it's a school, and being a school, there are, of course, lots of textbooks that can be useful to you. I'm presenting here things that are more basic from physics, from physical chemistry, the mathematics, and some examples for modeling. Plus, papers that are going to be cited over the classes. So yeah, feel free to take a copy. You also have available. And by the way, if at the moment, at any moment you need to get materials, just drop me an email, and I'll be happy to provide you for references. But this field is again, is an intersection of different things. So we are also touching things that belong to bioinformatics. I mean, we use the computers as our main tools. So as an example here, I may provide you basic textbooks for the simulation part. So structured bioinformatics can give you a general overview, and then we can move it to something a little bit more technical and more applied to different fields. One book here is in Portuguese. So I am also adding it because most of the people, they can talk in English, but I know that some are not comfortable yet. So if you are a beginner and would like to start with something that's more suitable to you, there is also a textbook here in Portuguese. Okay, you know the papers, you know the plan, you know the books that you can access. By the way, this one is for free. You just Google it and you can download the PDF. I mentioned to introduce something about myself. So I'm coming from the countryside of São Paulo State. We are here, is where people know the place where there is something called a pinguim. It's a beer place. So there is even a phrase in the country that if you come to Brazil and don't go to pinguim, you are not in Brazil. Sorry, guys. And I work for the School of Pharmaceutical Science. So what do we do? I mean, we try to understand the biomolecular interactions. So our point is, giving a protein, for instance, giving calcium ion, how do they interact? Giving a nanoparticle, how do they interact? A polymer, a protein-protein, nucleic acid, whatever. I mean, how do they interact? And in particular, exploring these things, salt effects, pH, and mutations, for obvious reasons. We work in three directions. One, of course, is to understand the interactions. The other is to develop computer models. Of course, we also use the models that are available. And one of the goals over the years has been to study virus and binders. Binders for therapeutic use, binders for prevention, binders for whatever you can imagine, the application, diagnosis, kind of things. So this, let's say, is a general overview of what we are doing and how we work in this field. Well, we are very curious about physics. So again, I'm back to my favorite example. I mean, these kind of things that I like to understand. So this is what motivates all the things. It doesn't matter if you're going to make an application to design a new antibody to treat a very important disease. But our main motivation is really the physics behind it. So this is the driving force for us. The general approach is like this, giving a very complex things. I mean, I'm taking here an example that's very useful to deliver drugs, drug delivery system, a microencapsulation system. Often you have a drug molecule that's going to be, that needs to be protected and is going to be transported and released somewhere. So doing so, I mean, you can improve lots of the pharmaceutical side of the problem, but when you look at it from a physical point of view, we try all the time to apply the spherical cowl trick or spherical bus. So it's a very complex thing, how I can simplify it and identify what is more relevant. If I'm able to identify what is more relevant and make a model to study it, I can learn from it. And once I can learn from it, I can start having ideas, improvements, these kind of things. So what we do here, for instance, for microencapsulation, was to identify that the key, let's say, part of this problem was to understanding the interaction between a protein and a polyelectrolyte. So this is, let's say, our call now. Well, how can we treat this? There are different options. And it's important to keep in mind that for each question that you are trying to answer, there is one solution, or there are a couple of solutions. We have to identify what is being relevant. And if you're trying to explore physics and to understand, it's better to try to simplify so you really know and have the full control of your system. And this is an example of what we do. I mean, we like to understand pH and salt effects, right? Together with the problem. So we create this very crude description of the system, our coarse grain models that look like that. So this is our spherical cows. I come back to more details. And using these models, we can get measurements that are comparable with experiments. For instance, titration plots. You have here the charge as a function of pH. This is a quantity that you can also get in the wet lab. We can get the free energies of interactions as a function of separation distance. So I can study affinities and understand what affect them. We can also get, if you're coming from physics, you probably heard about virial coefficients. So you can get the second order coefficient. So this is also something that you can do with such models. Now, again, this is why I was stressing that biomolecular interactions is important. If you're learning if you're learning biomolecular interactions, you can simply change your problem and repeat exactly the same trick. So I'm changing my slides, but I'm not changing the way that we rationalize the systems. Now I'm going to the immune system that's way more complex. And in this case, we do exactly the same thing. We identify the two molecules that are more relevant, and doing so, we are going to create a toy model so we can study how they interact with each other. And in particular, again, placing pH. When we are discussing antibody, pH comes again with more, let's say, importance because we also have pH that are controlled, antibodies that are controlled by pH. So in this case, I need something. And also, when you are trying to work what we call in bioinformatics on the large scale, I mean, you need to repeat your calculation for different mutants. You need to repeat your calculation for different physical chemical conditions, like you change the pH, you change the salt. So the number of calculations that you have to do is really big. You cannot wait 1 week per system. So you need to think about a way to simplify your model so you get the results in a quicker way. So that's why we have to think about how to simplify things. And doing so, we can get, again, titration plots, binding affinities, and in the case of antibodies and protein and viral proteins, for instance, it's important to characterize epitopes at the linear sequence and also at the 3D sequence. So this is an example. You see, I'm doing exactly the same that we did before for microencapsulation, but now I changed the problem, but I use the same tricks. And you can apply this in different things. I mean, I already gave you a couple of examples, but you could use this with food proteins. You could use the spidering problem that I mentioned to you, septins, and drug delivery like insulin, ketosome, this kind of systems. So, of course, I don't have time to go to all those examples, but I think I could give you at least a feeling what you can do. So let's go now to the very basic things. So now it's time to go to the fundamental questions. I would like to really try to address why we have this dependence of charge, this kind of things. So let's go to the basic physical chemistry. And when we start thinking about interactions, we know that there are 5 classes of interactions that we can have. In principle, we should be solving the Schrödinger equation. So it's a quantum chemical problem. But there are ways that we can make this in an effective way. In other words, we can make approximations that should work for the things. And in particular, we are going to focus here in what we call multipolar interactions, like this is charge, dipole, quadrupole, and so on, right? And also the induced interactions. If you would like to learn more about such things, there is a nice book here that I recommend you. So this can be a good starting point if you'd like to go into this business as well. So look into these things. I mean, why electrostatics is important? I mean, where do we get these things? Well, maybe instead of telling you from my experience, let's look at what people that got the Nobel Prize they used to say. And there are many examples. I mean, I'm just taking one older paper from Perutz. He got the Nobel Prize, if I'm not wrong, in '62. And in 1978, he published his paper in Science, where he started with this phrase, that electrostatic effects dominates, in some way, the protein behavior. There are many examples here, including something with virus, by the way. He already pointed out that this could be important for virus, as you can see here. So this is nice. I mean, we can see that it's important for different things. It was a Nobel laureate that was giving this information, but he was not the only one. I mean, you can see that in a talk that Ariel Warshaw gave in '98. I mean, this is a picture from him giving the talk, and you can see that again, for the structure-function relationship, it comes again to be mainly electrostatics. So, well, there are classical pedagogical examples. One is this protein that's responsible to transport the vitamin A, and in this case, it's CRABP1 and 2. They are, in terms of structure and the sequence. The sequence differs like just 30 amino acids, but one has a biological function, the other has not. And when we just overlap the structures, I mean, if you match the structures, you can see that they're very close to each other. There might be a loop here that they are slightly different, right? But in general, they are very similar. And in this case, you only see difference when you look to their difference in the electrostatic maps. So here is a nice example where we can see that the difference in electrostatic potentials change the function. So even if the difference in the sequence level is not enough to create a whole change in the conformation, it can be enough to change the electrostatic potential, and it can be enough to change the function, as you can see in this classical example. There are many others. I mean, I just pinpoint a couple of them for you. We could go to genetic therapy, and again, we are going to have the troubles because you have to understand how is the interaction of a polymer and the RNA so they can get in the cell and do the magic that they are supposed to do. There are many more examples. As I said, just a couple of things to illustrate why you have to listen to this basic physical chemistry. I often try to convince my pharmaceutical students that this is really important. And you can see here that it's the case. So you heard about the proteins. You heard about what is the composition of the proteins. You already have a nice class about this on Monday. I'm just trying to refresh your memory that proteins are everywhere. There are good examples. You can see this guy here. You can eat protein bars. You can have, I mean, a nice barbecue. I think everybody liked it on Wednesday. You can go further and you see that they are in cosmetics. They are even in toothpastes. A good example here. But probably don't know that there are even medicines using proteins like lactoferrin has already been available in the market to treat some virus, by the way. And it's from a physical point of view, you can imagine that it's just a collection of spherical beads connected to each other with different physical chemical properties. That's a way that we simplify things. But of course, it's not only like that. It's so important there is even a society for it and specific journals just with this name. So this is, let's say, one slide to summarize 3 hours that you probably heard with Chuck. But often when we're giving class for biochemistry, or we were describing proteins, we don't make very clear, or we don't go to this level of detail to explain why we have charges in proteins. I mean, you can understand most of the things, but often we don't let people know where do the charges coming from. I would like to remind you this, because this is essentially a very basic application of chemical equilibrium. So if you look at the amino acids, I mean, the building blocks of the proteins, we have an amino group, we have a carboxyl group, and you can have a side chain. But at least the amino group and the carboxyl groups, they can be titrate. They can have a change in their protonation states. So this is the source of the charges for a protein. And I'm writing down here the equations. I mean, the protonated state of the amino group and the protonated states are here. And you know that in this case, the charge is going from +1 to 0. So it's easy to see that when you look into the chemical equations, the charge can vary for every amino acid that can have an amino and a titrate, amino or carboxyl group. Of course, when you make a protein, you have the protein, you have the peptide bonds, and you're going to lose great part of them. But even so, you are left with some side chains that can also have titrable groups. In the case of the carboxyl group, you can see that the charge here can vary from 0 to -1. So this is the source of the charges. And pH is the one that controls such things. When you are in a low pH conditions, like you have here in the picture, the concentration of the protein in the solution is quite high. So the protein is happy to be present in all the sites that it can be. So you have +1 here, so the amino group is protonate, and the carboxyl group is also protonated. So the net charge of the single amino acid in an acid condition is +1. When we start increasing pH, it means that we are decreasing here the concentration of the proton. So now, where the proton is not so happy to stay, and if it's missing the solution, the proton decides to give up and go to the solution. That's what happens with this carboxyl group when you are at physiological pH, pH 7. So in pH 7, you have now one group that's protonated, the amino group, and one that's deprotonated, -1. So now I changed my charge of this single molecule from +1 to 0. And if we go further to the more basic regime, we end up in this condition that even the amino group is going to give up its proton to the solution. And now I have a negative molecule. So for a single amino acid, I could change the protonation state from +1 to -1, simply change pH. And the thing can be more complex because, I mean, come on, I'm talking about one amino acid. I didn't even talk about the side chains. But what we are doing, I mean, so let's look a little bit to the equations. And in this case, being a chemical equilibrium, we can write the chemical constants here in terms of activities. Activities are the true thermodynamic quantities. So we can simplify them and look into the ideal conditions. And in this case, we change the activities and make an approximation to look into the concentration stats. Now we have a stoichiometric quantity. So this is a simplified version. I mean, when we are putting aside the interactions, we are assuming an ideal system. But remember that case can be very large or very small. All the time that you have to deal with large or small numbers, it's very convenient to work in a logarithm scale. So that's why we have pH as a definition of minus log of the concentration or minus log of the activity, because we are dealing with large numbers. We do the same here, and often we work with pKa's. So that's the beginning of the pKa's, or that's the definition of the pKa's that you heard since Monday. Another advantage of working with pKa's is the fact that when you can directly relate them to the free energy. So you can see if you just replace them by the definition here, if I increase the pKa, I increase the delta G. So they goes in the same direction. It also helps you to rationalize when you are doing applications, because it goes in the same direction. So it's also convenient. And if you'd like a phrase to describe in a simple word what we have for pKa, you can see here that pKa is simply a measurement of how easy or what's the tendency for a group to give up a proton. That's what we are measuring in these pKa's. Okay, that's the initial definition. I mean, from a historical point of view, the first definition happens to be at Carlsberg in Denmark. They used in that time the concentration, and it was just later, probably, I would say, from my opinion at least, it was thanks to the Biomolecular that they have seen that it was activity, the better concept. And now we have, let's say, this simplified version and this more advanced version here, more modern version. So when you look into the building blocks, I mean, we have also talked about the side chains. And the side chains, they can have different physical chemical nature. A couple of amino acids can be apolar. Others happen to be polar. And then we have the ones that are acid and basics. And the ones that are acid and basics, they can have titratable groups, as you can see here. So you have a carboxyl group for the aspartic acid, this another one here for the glutamate acids. And also we have groups here that can also work as an amino group with a positive charge. What people often forget is that cysteines, when they're not involved in sulfur bridge, SS bridge, they can also titrate. They can also lose or gain a proton. And also tyrosines that are here. Okay? So these are the groups that can titrate, and now you can imagine that, I mean, all these effects are going to be together. Just to try to help you with this, let me just go a little bit to a couple of more slides, and you can start looking to the numbers that people were measuring. Here is one of the initial data. I mean, you can see the chemical groups and their different protonation states and their pKa's. One guy that has been my hero in science was John Gamble Kirkwood. He has a book, by the way, called Proteins, and everything that I'd like to know about proteins, it's there. Whatever you have an idea to say, well, I'm going to start a new field because I have a very clever new idea. You go there and you can see that he has been already working for this. Maybe, thanks to the progress, he was not available. I mean, some information was not there, and you can still have to do. You still have the opportunity to do some work. But Kirkwood has probably at least scratched most of the more important proteins problems that you can have in proteins. And he was working very close with Lindström Lang, that happens to be the director of the Carlsberg Brewery in Copenhagen. So whatever you drink at Carlsberg, you were at least in that time contributing with science. It was not a German company, by the way. Sorry, guys. Anyway, having the pKa's, it's a simple way to calculate in your pocket calculator or by hand the degree of ionization and the net charge of individual molecules. So we can see here, for instance, that you plug the pKa, you know the pH, you know the degree of association, and you calculate the charge of both a basic or an acid amino acid. And here you have an example about the titration plot for an amino acid, where we have as a of a pH, and when it's half protonated states, we have what we define here as a pKa. So you have an illustration of the pKa, at least a general scratch of it. Is it clear for you? I mean, I guess most of the people knew about these things, but maybe there are others that are not familiar, so I'm trying to make sure that we are on the same pace. But there are things that even if you know about this, we always forget. We always forget about what I call here the neighborhood effect. So if you look at a single amino acid, now I'm back again to a single amino acid, and in this case, I'm taking two amino acids that are quite similar. I mean, both are acid amino acids, glutamate and aspartic. The difference is that the glutamate acid has this extra CH2 group here, and if you compare their pKa's, even when they are isolated, you can see that they are not the same in numbers. I mean, look here, the carboxyl group is 4 in aspartic acid, and here is 4.5. So why they are different? Well, they're different because these groups can communicate. I mean, they are coupled to each other. So you can expand this idea and understand, I mean, why in a 3D structure of the protein, when you change the protonation state of a given side chain, it can also affect all the neighbors. Sometimes it's a short communication, it's a short process, just the local neighborhood. But sometimes we have what we call a high coupling effect. So I do a change here, and it's going to propagate to different titratable groups until amino acids that are far away from the interface. That's something important to keep in mind, because we all the time have this lock and key mechanism in our mind. And when you're talking about electrostatic interactions, if you do a change far away, but you have this high coupling electrostatic mechanism, that change very far can affect my interface, and is going to interface how I'm going to interact with my partner. So this is important to keep in mind. You can see this in proteins. Yes, please. It's just like when you change one protonation state, you, of course, the other one, if it's that close, is going to feel that you change the charge. Yeah, it's like they go like in a cascade effect. I change this one, I change the next, and then eventually I change the next, the next, the next, the next. You can really map how this is done. Okay. When you look at your 3D structure of the protein, you can see here, for instance, we have ASPs, I mean, aspartic acid in different positions. I mean, this is just their position in the sequence. And you can see, depending where they are located in the 3D structures, their titration plot is shifted. So this shifted is the effect of the neighborhood effect. So actually, when you're trying to understand the electrostatics of proteins, or in proteins, you have to look very careful to what happened with the different titratable groups. So you can see how is the impact of the 3D structure in their titration properties. So this is just one example. You can use this information to predict what we call the ideal charge of the protein. And that's something that you can also do by hand, because you can simply know what happened with individual amino acids. You know the number of that particular amino acid in a given structure. You can just add up everybody together and you have the total charge. Look here, I'm just using the pKa's to get these numbers, as I showed in the previous slides, which means that 3D effects are not there. We are looking to the effect of a single amino acid only. And you can see that there are differences. I mean, you can see, for instance, here, for lysozyme, the ideal behavior in dashed line, and in solid line, we have what you are going to quantify if you use the 3D structures. But there is something quite nice that around the isolectic point, where the net charge of the protein goes closer to zero, we can see that these predictions are quite good. So, in fact, there is an old paper where they are comparing experimental PI results with theoretical ones using only the sequence, not the 3D structures, and the correlation is not that bad. I have done some exercise on this as well. I mean, you can see here for a couple of proteins, when you compare this ideal behavior with Poisson-Boltzmann and experimental data, that in some case, the results happen to be exactly the same. So you can get exactly the same pKa's, for like calbindin is an example, this is a calcium-binding protein, and you get exactly the same charge if you were working with ideal sequence level, or you're working with the 3G structures. In some cases, actually, it's even better, the ideal result. I mean, ribonuclease is a good example. You get 90.4 here for your RMSDs, and the experimental number would give you 90.6. Oh, sorry, for the RMSD. So it's, I mean, it depends what the protein we're talking about, but you can get a good estimation of the isolectic point just using the sequence. That's why codes like XP, whatever it's called, one of these tools that you have in the Swiss programs, available tools, you can have a good estimation of the PI. Okay, so another thing that we have to remember that PA controls conformation. So for instance, if you neutralize these guys, increasing the pH, you can see that you're going to decrease the repulsion, and eventually you get an attraction here. So you can change the protonation states, you can also change the protein conformation. And as you know, I mean, from biochemistry for sure, the pH is going to control function. You can see that different enzymes, they have their optimal pH conditions where the activity is better for that particular proteins. And the numbers are different. These are just 3 examples that I pick up from a biochemical textbook. So pH controls function. I guess it should be clear at this stage that pH is really very important, right? I mean, you've got lots of examples about this. To summarize the whole thing, I just bring back this equation that the charge of a protein is a function of the pH conditions. And there are, of course, other details, as you can see here. When you change the conformation, I mean, changing pH, you change the conformation, you have a chance to expose more hydrophobic amino acids that like to be inside the protein. So you can gain extra in terms of attraction because you can get this attraction thanks to hydrophobic interactions as well. So there is something else that we needed to discuss. I mean, I talked a lot about the pKa's for the ideal molecules. But where do we get these numbers? I showed you the Lindstrom-Lang data and Kirkwood. So these are the same ones that I'm reproducing here again. But are they all? I mean, do we have more experiments? I mean, this paper is more than 50 years old. What about recent data? Well, this is the classical one from Tanford and Ozaki. They are the ones that measured. It's often the ones that we have in most of the simulations, the Constant-pH simulations, they often calculate what we call the pK zeros. So for the model complex, for the ideal conditions, you can already see that if you change the salt concentration, for instance, or the environment, you change these numbers. Look at the blue, the green, and the red here, and you can see the numbers are different. And in fact, if you look at the collection of experimental data, you can see more diversity. Different labs have been measuring these things. And the main difference between each of these measurements is the peptides that they use to calculate it. So you can see that sometimes they are using pentapeptides, or sometimes they are using tetrapeptides. X here is the group that you are trying to characterize in terms of electrostatics. Again, you saw a comparison between ASP and GLUE, where I have an extra CH2. And that group was enough to change the protonation states of my amino and carboxyl group and side chains. If I change the length of my peptides, so my neighbors, of course, I'm also going to change the protonation states. I mean, there is a chance that this is going to happen, right? Because if you solve for something simpler, when it goes to more complex, it's expected to see. And this is the reason why you have a set of different experimental data. So depending on what application you're doing, it's good to keep in mind that this phenomena is also going to be present. And it's one thing that you have to take into account when you're doing your constant-pH simulations. You have to make a choice for your input pKa zeros. Often, we keep going with Ozaki and Tanford. That's the one that you have here again. It's the same that you were there before. But there are new data, and there are even new experimental results where, for the first time, this is about 20 years ago, they finally were able to estimate even the error bars of their measurements. This was a nice achievement, because then we can see a little bit better how they are really, I mean, what do they mean. But this is, let's say, a recent result. These ideas, I mean, we can use it to predict, for instance, so now I'm going from the basic physical chemistry to a real application, but using exactly the same ideas that we can see in basic science. So we came up with this ProCDPKA method that happens to be just a way to identify the most relevant amino acid for an interaction between an antibody and a viral protein, for instance. So what we use it here is that to try to map where are these binding spots, we use this idea. I'm placing here a molecule, and in the presence of this molecule, I have a neutral protein, a neutral charge. So we know that if I approach a positive charge, I'm going to affect the protonation states of this guy. This is the neighbor effect. And in fact, you can even quantify it. If I put +1, you can do a simulation. You can see that the pKa of this group, that was 12, is going to go down. I mean, you have a shift on the left direction, and you have a difference here of 0.7. If I apply this for a polymer, so I have a combination, a polyelectrolyte, a combination of charge that's going to be attracted by a protein, this polymer is going to visit different sites. So what we have in the case of a polyelectrolyte interacting with a protein is that not just a single charge, but a collection of charges from the polymer might affect the protonation states of the group. So if I know how is the behavior, the titration behavior of the protein isolated, and then I do the same analysis or do the same calculation in the presence of the polyelectrolyte, and again, I have the pKa's in one condition, I have the pKa's in another, I can see the difference. And if I compare the difference, I can identify where the polymer has been more often. Because where the polymer has been more often, I probably have to see large shifts in the pKa's. So if you do this for protein polyelectrolytes, you can get maps like these ones, where you can see in blue the shifts that were positive, and in red the shifts that were negative. So now we know that for that particular example, that the polyelectrolyte has been visited more or less all the surface. It's not just like in a patch, it's visited just one area, but it has been explored all the regions. So this is an example of a non-specific interaction. You can also have an application where you can see more specific interactions. So all these ideas were incorporated for epitope prediction. So now we know for a given antibody where are the regions that are really relevant for in the antiviral protein or the viral protein. Okay? Is that clear? Sometimes I look to you and you have a question mark face. Okay. Okay, I'm going on. I think just to remind you about the experimental part for the electrostatics, I'm going to cite some examples, some techniques that are often used from more macroscopic ones to the microscopic ones. I don't think I'm going to go to the details. My time is, of course, running, but this is just to give an example to you. And there's a nice review paper 25 years ago where you can find it. From the theoretical point of view, everything starts with this paper from, again, Lindström Lang. I hope you can read Danish. Otherwise, Rolf might be able to help you. And this is the first time that someone was talking about ionization of proteins. Of course, Google can help us nowadays as well, but you don't need to read the papers, just like it was just the basic ideas. And from a historical point of view, we have a couple of landmark papers. Like we have their paper from 24, then we have the Kirkwood mathematical work that was the basis, the mathematical basis for the Thunford-Kirkwood model that I'm going to describe it to you from 57. Then we have the first Poisson-Boltzmann simulation for a protein. That's from James and Warwick in 82. We have the first Monte Carlo simulation for a protein. That's from 80 years later. And then we have the beginning of what we call the molecular dynamics or the constant-pH molecular dynamics simulation, thanks to Antonio Batista and Peterson from 97. In 2011, it was the beginning of the simulations with, or not simulations, but the empirical methods. This used to be my slides until 2020. Now, we probably have to expand it because we are in the machine learning area, and there are many new models that are appearing. I mean, just to give you an example to you, I'm citing 3 new methods. By the way, with a curiosity, because Machuqueiro happens to be a former PhD student from Antonio Batista, so they are coming out with PKA methods or electrostatic models, let's put it like in a more general way, that are using machine learning in different ways. So you can see that this has been expanded a lot. We are also working in a similar model like that, but this can give you a feeling, I mean, how we can evolve in the field. Often, when we are developing methods, it depends how would you like to represent your call. I mean, all the time you have to see, I mean, what is your question, first of all, and how is going to be the balance here between what simplification you can make, what accuracy, and what practical relevance you have, what is your computer power. I mean, you have to put everything. So there is not really an ideal model for everything, or a perfect model for everything. For each of these specific conditions, you might have one model that's better for your own application under the condition that you like to study. So keep this in mind, because we can select the call that you prefer depending on applications. They are all the same animals, but sometimes this one can be good enough, or maybe this is the best that we can do with the available resource. Sometimes we go to a real one. I mean, it's up to you depending on the applications, right? To try to illustrate a little bit it, before we go for coffee, I'm just reminding you here that there is a nice paper, very old nowadays, where you can classify the main theoretical approaches. And in this case, you can see here that, of course, for a given physical system, we can use our models, I mean, our force fields, if you prefer this word. We can go to the quantum level, where we define particles by nuclei and electrons. So this is a way to describe things. But unfortunately, this is very expensive in terms of computer resources. So often, we have to invoke simplifications. One of the simplifications is to go to the next level, where we have the microscope level. And here, these people call here the Born-Oppenheimer level, where we describe particles and define them as the solute and the solvent. Here, we have a split solvent. But come on, for some applications, this can still be too costly. So depending on what you are doing, you can go one next level. So we can go to the macroscopic ones, where now we get rid of the explicit water models. We just use the average behavior of water. And we go in a class what we call implicit models of solvents, or continual description of the solvent. If you'd like to go further, for some reason, we can also go to phenomenological descriptions. That's also possible. So this is just a couple of things. So depend what you were after, there's one that's better for you. Just keep in mind that the CPU costs increases when you're going from the left to the right. Okay? Questions so far? No questions? Okay, so let's go for a break. Rolf, what time would you like me to come back? I think 10 to 12 could be fine. Like, yeah, we have a long day, right? Okay, so we are back 10 to 12. Okay, thank you. Oh, thank you.